On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Menarini Silicon Biosystems, I'd like to welcome you to Utilization of DEPArray in Molecular Pathology for Clinical Diagnostics and Translational Research. My name is Elizabeth Lamb, and I'm the host and moderator for today's event. Now, I'd like to introduce our presenter for today. He is Dr. Matija Snuderl, MD, Associate Professor, Department of Pathology, Director, Molecular Pathology at NYU Langone Health. Welcome, Dr. Snuderl. The presenter ball is yours.
Thank you, Elizabeth. And thank you all for attending. Good morning, evening, or afternoon, whichever time zone you're located in. I look forward to present to you our results in using DEPArray both in the clinical and translational research setting. The webinar is styled towards practical applications, critical bottlenecks, and whatever you need to know if you're thinking how to set up your DEPArray experiments. And I look forward to your questions. So what we'll be talking about is what is actually the role of DEPArray sorting and cell selection in clinical and research molecular pathology. I will review with you critical steps in the DEPArray workflow, focusing particularly on cell suspension preparation, cell staining, how to perform a successful DEPArray run, and then talk about cell collection and volume reduction, molecular analysis, and how to implement DEPArray in either clinical NGS workflow or how to utilize DEPArray in translational research and cancer heterogeneity. So DEPArray technology comes in a crossroad when you're thinking whether you want to perform both tumor sequencing versus pure cell population analysis. And there's no question that a vast majority of laboratories perform simply both tumor sequencing because it is easier, simpler, the workflows are very well established, and we know what our limits of detection are. But there is also a point where we need to discuss pure cell population analysis, and this is going to be the topic of today. The first point that I would like to emphasize, although I'm sure that everybody on this call is familiar with this, is that cancer cells do not live alone in the tumor. There's many other cells that coexist with cancer cells and end up diluting your genetic information as you're performing your sequencing. Among them are, for example, endothelial cells, which are showed here as an exuberant microvascular proliferation with red arrows, or immune cells, highlighted here with a stain for CD163, showing a tumor-associated macrophages. And you can see about 30–40% of cells present in the tumor are actually tumor-associated macrophages. The quality of NGS results highly depends on tumor cell percentage. And this is just an example of one of our clinical validations when you can see a set of mutations. The font is probably too small for you to read, but is a bunch of single nucleotide variants and small insertions and deletions at the, I would say, lower limits of detection between 10% and 2.5–3%. And you can see the detection at no dilution and then how the detection decreases as you're diluting your specimen. In addition, you can see that your ability to detect these mutations also decreases with decreasing DNA input. While your decreasing DNA input may not have much impact when your sample is not diluted, more you're diluting in, higher the impact of your decreasing DNA input. This has effect not only on tumors on variant allele frequency that you're calling, but also on the coverage, which is again highlighted here, that with no dilution, as you're decreasing, the smaller DNA input has a relatively small input. Here you see dropping out one of the known mutations, but more you're diluting the sample, more dropout you're going to have in your coverage in mutations that you're interested in. And this has been shown by many studies, and it's just to reiterate that there are two factors when you're setting up your sequencing experiments: tumor cell percentage and DNA input. Even more critically, I would emphasize, is that copy number calls are highly dependent on tumor cell content. And you can see the experiment here we set up that shows you samples with various amplifications of various genes and then being sequenced and called copy number amplifications. And you can see how rapidly the number of copies called by next-generation sequencing decreases as you're diluting your specimen. While tumors that have a very high level, starting level of copies, do relatively well even with the significant dilution 1 to 10 or 1 to 20, The genes that are amplified with a relatively lower number will be very much affected by your dilution, and you're not going to be able to call it at a threshold which is here established at 5 copy numbers. So this represents a practical problem that small and low tumor percentage biopsies are challenging for clinical next-generation sequencing, and poses the question, how do we isolate the pure cancer or stromal cells for research purposes from FFPE? FFPE material is challenging for traditional single-cell techniques. Always when we submit a grant using DEPArray to NIH, there is always somebody who is smart and says, oh, why don't you just use 10x genomics? And we try to explain them that traditional single-cell techniques that are possible on a very high-quality fresh samples are simply not amenable for FFPE material. Slide scraping can improve your tumor cell percentage if the normal and tumor are clearly demarcated, but the intermixed tumor cells from the tumor stroma, as I showed you with endothelial cells and immune cells, cannot be done. In theory, laser capture microdissection can be performed, but that's very time-consuming effort and not really feasible in clinical workflows. So the implication in practical life is that stage 4 cancers, or patients with stage 4 cancers, who are often not candidates for tumor resection and get a small biopsy or an FNA, and that might be the only sample that's provided for the testing. So the paradox is that the sickest patients who would benefit the most from next-generation sequencing or other molecular testing usually have the smallest samples. While if you have somebody with stage 1 lung adenocarcinoma and you get a total sample resected, that's often not indicated for advanced molecular testing. So what do we do with it? So there's some practical considerations that we need to keep in mind, and that is that most clinical NGS panels have limits to detection set about 10 to 20% of tumor cells in the sample, and that varies between assays and laboratories, but I would say that 10% of a tumor sample is about as low as we push it in a clinical laboratory. And the result of that is that single nucleotide variants and indels, variant allele frequency calls, are limited to either 5%, rarely less. Some institutions call hotspots down to 2 or 3%, but that's becoming a little bit complicated because you're dealing with a false positivity ratio. So let's say that our threshold would be 5% and 10% of tumor cells, which means if there's a heterozygous driver mutation, which would be present in 50% or 50% of your tumor reads, at 10% tumor cell percentage, you'll be able to detect a 5% call. Copy number detection is significantly impaired once you go below 50% of tumor cell content, and I would say the detection of homozygous deletions is not reliable under 50% of tumor cell content, and I will show in one of our experiments how DEPArray can help with that. And then the second thing, as I showed you, is we often talk in clinical laboratories about tumor cell percentage, but it is also DNA input that plays a significant role. You have to have a biopsy or resection big enough to provide you enough of a DNA input. And that ranges between about 10 nanograms for amplicon-based NGS chemistry to about 50 to 100 nanograms for hybrid capture. And again, it depends on the laboratory and the assay. In our clinical experience, about 10 to 50% of cases that need clinical NGS is below these levels of detection. Either there's not enough tumor cells or the tumor sample is too small. And that, of course, depends on the practice and the type of medical center, but that is approximately experience around the field. This often affects small biopsies, fine needle aspirations. And insufficient biopsies are particularly problematic, as I said, for patients with disseminated cancer who are not amenable for large resections. And in addition, lower tumor cell percentage also affects your ability to detect subclonal events. So it is nice that at 10% tumor cell content, you can detect the major driver mutation that is present in every single tumor cell, but you will not be able to detect the minor subclones or resistance mutations. In addition, tumor cell percentage is not the only issue that you're facing. And in our active clinical and translational research, we are dealing a lot with the tumor heterogeneity. And as you're all aware, tumors are not homogeneous. This is just an example of astrocytomas, a little bit simplified view of how astrocytomas and high-grade gliomas develop, but you have some early mutations like IDH1 and 2 mutations, ATRX, P53. Then you have a later mutations, such as PTEN loss, amplification, and then in the secondary, like later on, GBMs or primary GBMs from the beginning, you have amplifications of receptor tyrosine kinase genes such as EGFR, PDGFR alpha, or MET. So all this results in having a very heterogeneous population of tumor cells that you have to deal with and take into account when you're profiling them. We've shown now several years ago that mutations and copy numbers can be present in the same or different cells. So if you look here, you can see that amplifications of genes such as EGFR and MET in this tumor is present in mutually exclusive populations. Cells with EGFR amplification do not have MET amplification and vice versa. But in this tumor, amplifications of MET in green and PDGFR alpha in red are present in the same tumor cell. In addition, you can have multiple mutually exclusive subclones coexisting in the same tumor. EGFR amplified tumor cell, PDGFR alpha amplified tumor cell, and MET amplified tumor cells all coexisting together. And this is something that you can do with analysis in situ very well, but you're of course limited with the number of probes and resolution that you can do in situ analysis. So the goals that we set on doing is how do we isolate pure populations of cancer cells for accurate molecular analysis? And in addition, how do we identify tumor cell subclones and subtypes to be able to better dissect tumor heterogeneity? There are other webinars on details on DEPArray technology, so I don't want to spend too much time with this talk focusing on that, but just for those who are new to the technology, I want to emphasize that DEPArray is basically a combination of electronics, microfluidics, and microscopy. The sample gets injected into microfluidic channels, comes into the chamber, is visualized with a microscope and a camera, and then the cells are analyzed, looked on, and then moved the recovered part into the tubes for further molecular analysis. When you look at it a bit more closely, it is called the array because there are these microelectrodes that are in the bottom that you can basically switch on and off, and you can create this negative field surrounded by positive fields. So when the cells get trapped in them, you can move them around with switching the fields. This is a cross-section of the DEPArray cage. Because you have a negative field surrounded by positive fields, the cells will drop into the field and be caught in there, which is a process that is very gentle, and with the switching of the pluses and minuses, you can automatically move the cells in any directions you want. So this way you can capture multiple cells, you can visualize them, decide which cells you want to pick, and I will show you an example of that, and then move them into direction and recover them. I want to spend a little bit of time on the DEPArray workflow. We'll be discussing cell suspension preparation, staining, DEPArray around itself, but only briefly, and then cell collection and volume reduction, which are some of the new things in the DEPArray workflow, and then molecular analysis, which is the most important. So when we talk about cell suspension preparation, of course, every good experiment starts with a good sample preparation. Starting material is one FFPE scroll. A recommended thickness is 16 micrometers. This is a thickness that has been working very well for us, but you can decrease the thickness or you can increase the thickness. Decreasing the thickness, please note that you have to keep in mind that you're trying to keep the whole cells. We've done some experiments. You can take a 20 micron scroll, you can take a 10 micron scroll, and that is all possible, but you will lose the benefit of a whole cells, and I'll show you why that is. When it comes to the large size of cells, I think the theoretical limit of how big cells you can push through the microfluidics is about 100 micrometers. But it depends not only on a single cell how big they are, but cells, even the best suspension protocols, will still leave you with some clusters. So if your cells are large, 60, 70 micrometers, and you have a cluster of them, they may clog the microfluidics, and you will not be able to see cells. But this would be about a theoretical cell size that we were able to recover. For the cell suspension preparation, you can use the DEPArray FFPE sample prep kit, which is very simple to use. Enzymatic dissociation, has all the enzymes pre-mixed, and contains two antibodies, anti-cytokeratin and anti-vimentin antibody, and then a critical FFPE QC kit, which will help you to estimate the number of cells that need to recover for successful molecular analysis. I would say that the QC kit is very conservative. We have gone way below the requirements of the kit, and we're able to get a good sequencing analysis, and I'll show you that in a moment. For practical consideration, keep in mind that this is sort of like one-size-fits-all kit, but these different tissues may require a little bit of different sample preparations and dissociation protocols and timing, and you may want to experiment with different enzymes such as pepsin, lipase, or papain. We have tried them. Papain works very well on the brain tissues. Lipases are useful when you're dealing with the very fatty tissues. And pepsin has been working in some tumors that had a lot of elastic fibers. So a good starting point is to start with a sample prep kit and then modify based on the experiments that you want if you're not able to achieve ideal suspension. And having really a single cell suspension is a critical piece to be able to get a pure cell population. So why am I obsessing so much about the whole cells? It's something that I call the original sin of a 5 micrometer unstained slide. And everybody who practices either molecular pathology or is getting samples from the clinical laboratories is probably aware of the fact that standard pathology cuts slides for molecular analysis at 5 micrometers. When you think about it, just this kind of like a example of a cell nucleus. Is a cell nucleus is about 20 micrometers, I would say, on average. But of course, it ranges. Large tumor cells can be, 100 micrometers or more, and variability is huge. And when you cut these cells to put them on slides, you're basically getting on every slide just a fraction on all of each nucleus. So at the end, you're ending up with a standard slide which has just like 5 micrometer thick portions of the nucleus, and that means only a fraction of the DNA for from that particular cell. And of course, you have a lot of cells, but you have only fractions of each of them. And that is actually the reason why clinical molecular assays require a huge amount of DNA to overcome this problem that from every nucleus you have only a fraction. So you have to put a lot of fractions together to be able to reconstitute the genetic information that you're needing. So the hypothesis that whole cells are better than fractions of the cells is that you'll be able to not only get a pure cell population to analyze, but you'll also be able to significantly decrease your starting DNA input without negatively affecting coverage. The second step is the staining. Staining is the trickiest. While the FFPE prep kit from Menarini Silicon Biosystem is very straightforward and it can be used in any laboratory immediately, it contains only 2 antibodies. One of them is a cytokeratin and the other one is vimentin antibody. There are very few directly conjugated antibodies that are optimized for FFPE. So one of the approaches that you can do is you adapt your primary antibodies and you basically replace cytokeratin and the vimentin antibody in this kit with antibodies of your choice, as long as the secondary antibodies from this kit will react with your new primaries. So that is absolutely possible. And here's an example of adapted CD163 antibody and a GFAP antibody that, in the same APC channel that is originally designed for vimentin, we used to be able to detect microglia and astrocytes. I would recommend, from the practical perspective, start with antibodies that work well on FFPE and then work from there. Do not start with antibodies that work well only on flow or only for Western blotting, because you're at the end of the day, your tissue that you're dealing with and your artifacts and antigen retrievals are still FFPE. So that would be my recommendation for that. So you can use a different primary antibody as long as you use the same secondary antibody. But that's not always possible, because the secondary antibody from Menarini will recognize only some of your primaries. So eventually you'll have to design new primary and secondary antibody combinations that will fit your project. And in that aspect, you can see an example of the sort of isolating or different cells that we did from the brain. Here, there's an example of cortical neurons that stain in APC for MAP2. Forgot to label here, this is a FITC for NUN. Astrocytes, they're staining in GFAP with GFAP and FITC. Endothelial, brain endothelial cells, strongly positive for GLUT1 and FITC channel. And here from the same experiment, macrophages that are staining with CD163 and APC channel. So you can eventually down the road build a library of stains that are going to be working for you, so you can isolate the cells of interest in your laboratory. The DEPArray run, I will not dwell on it too much. It's fairly automated. Particularly the new VRNxT system has significantly simplified cell selection. So it is almost completely hands-off, Once the technician or student or postdoc load the chip gets scanned, and it helps you automatically select the cells that fit the criteria, that are in a cage, that are possible to be rounded and possible to be moved around. And then you can select your groups of interest using the intensity from your staining. You can also analyze the morphology. The system allows you to look at multiple different parameters to assess the cell nucleus, size, shape, and everything. You can analyze also ploidy. And then you park and recover cells by dropping either into PCR tubes, 96-well plate, or directly on a slide if confirmatory studies that you want to perform by our FISH. Cell collection and volume reduction is probably the most critical step, particularly in the beginning. It is very concerning when you set up a beautiful experiment and you collect the cells in a plate or in a PCR tube, and then somebody with shaky hands aspirates all single cells or 5 cells in the tube out of the tube, and then there is nothing. As everybody knows, volume reduction by manual pipetting is extremely user-dependent. It requires very good pipetting skills, and you may have technicians that are extremely skilled with pipetting, but that requires a lot of training. But if you're like me and you have a lot of students and postdocs who rotate through the lab, and it takes some time to gain those skills to be able to really perform volume reduction well. So about last year, we learned that Menarini Silicon Biosystem will be launching this new tool, which it's just called VRNxT, and that allows you to efficiently automate volume reduction, streamline and standardize your traditional manual steps in the workflow, and significantly reduce the error. We participate in a beta testing of the machine to see how it actually performs in clinical practice. And I don't want to dwell too much on details, but you basically pellet cells at the beginning, then you put it into VRNxT, and what happens is the center is for the rotation is pulled right above the cells, about a 2 microliter volume. It rotates it around, and that basically leads to the waste to be pushed out of the tube, while your single cell or small numbers of cells are pushed to the bottom. And it's a very fast process. It holds 4 samples, and it's done in 7 seconds. The only disadvantage is that you have only 4 samples that you can put in the machine. But it's still much faster than doing it manually. When you look at the performance of the machine versus different users, you can see that the machine is very consistent. It has a significantly lower error rate. It is, most importantly, much faster. There's a huge variability among the users, and these are all experienced users. But if you imagine putting sort of like a virtual student that is for the first time pipetting, done maybe 100 pipetting at a time, their performance will be much lower than this. So the most important is that you can recover your cells with a very high accuracy and very quickly. So now, let's move towards the most important part of this discussion, and that's the actual molecular analysis. So I will discuss with you 2 possible ways how you can perform the analysis. One of them is you don't have any sequencing capabilities in your laboratory, and you want to do everything out of the box with Menarini kits, and that is possible. The OncoSeq panel and then whole genome amplification kit with low-pass whole genome allow you to analyze the cells that you've recovered. There is now a new feature which we have not used yet, because we rely on our pipelines for now, using automated bioinformatic analysis of the sequencing data through a tool that's called MS BioSuite. As I said, we didn't try it, but that's one of the tools that's available if you don't have a stronger bioinformatics group. And then the most important is most of the laboratories already have some sort of next generation sequencing panels established. If it is a clinical panel, you spend a lot of time and effort validating it, and you don't want to just grab another sequencing panel and revalidate it. So can you use it? So we tested DEPArray extracted cells by 2 clinical panels, our hybrid capture chemistry, as well as an amplicon NGS assay. I'm going to show you examples of one of them. So let me first show you the proof of principle study. This is sort of a classic scenario that you would choose to show that the system works well. This is an infiltrating astrocytoma. This is a mutation-specific antibody for IDH-R132H mutation, and this tumor is sequenced. You see, have a relatively low tumor cell content in here, and this is a paired blood. And you can see that we are indeed able to detect this mutation, although with variant allele frequency about 7%. You can see there are no mutation calls present in the blood. As a proof of principle, we capture with DEPArray tumor and stromal cells. For the tumor cells, we identify them using IDH mutant R132H antibody to show that we have a really pure populations of cells. And you can see that our variant allele frequency jumped to 47.5%, which means we have almost pure population, probably pure or almost pure population of only tumor cells. And here you see in GLUT1 endothelial cells from the same cell suspension that we are using as our matched normal. The reason we are using this and not the blood is to show that that we have a good specificity, and then we call something a tumor cell, it is indeed a tumor cell, and when we call something a stromal cell, it is indeed a stromal cell, and we don't see any contamination of our endothelial cell pool with potential tumor cells. But you can say, well, why does it matter? You were able to detect it at 7% before. What is the point of going through all this extra work just to prove that you can do it better? The reason is that this mutation is an early driver event. It will be present in all the cells of the tumor. And because we had a low variant allele frequency in the first event, it means that if there are any subclonal events that are present in a smaller fraction of cells, we will not be able to detect it at our clinical threshold of 5%. And this is example from that same case. You can see that the 23% variant allele frequency after DEPArray capture, we are able to detect PIK3CA mutation. And estimated variant allele frequency, and we actually saw it in the reads when we went back, but did not pass our clinical threshold. Estimated variant allele frequency is about 3.3% in non-sorted sample. Because our clinical threshold is 5%, we would not be able to call this mutation. And this is a significant mutation, and that we have it now, we can call it because it predicts more aggressive behavior in gliomas with IDH mutations. So by having a pure population of cells, not only that you can better detect the drivers that are dominant drivers, early drivers, but most importantly, you can detect subclonal events that you would otherwise miss. So how do we implement in clinical NGS workflows? We focus particularly on small samples that would give us a low DNA yield, and then samples with low tumor cell content. I'm going to show you examples of our clinical accuracy studies and reproducibility studies. So this is one of the samples that we use for the clinical validation of the DEPArray, using the DEPArray FFPE sample prep kit with a FITC for cytokeratin and APC for fermented. And you can see gating here on the tumor cells in FITC, on the stromal cells in APC. This is your cell browser. You can see the tumor cells here that are highlighted and selected have a very strong expression of FITC. And then when you do your recovery of cells, you can see it basically gives you this 96-well plate, and it tells you, and this is just dropping before you recover the cells. This is the group 1 of tumor cells. Each of the cells carries a specific barcode. Of course, when you're dropping them as a pool, that is not that particularly important, but once you start thinking about a single cell analysis, because you know for every PCR tube what is the barcode of your cell that you dropped in there, you can then go back to your images and compare, let's say, your low-pass whole genome or mutation information for every cell that you drop with that particular image, and then you can start doing some interesting analysis on how morphology and genetics interact with each other. So what was the clinical characteristic of this cell, of this sample before we did DEPArray? At the original tumor cell content was estimated from H&E and 30%. We detected from the bulk sequencing 2 EGFR variants, exon 21, L855R, and T790M mutation. You can see variant allele frequency about 20 and 23%. So we are a little bit off with the tumor cell percentage estimate. But when we do DEPArray, you can see these are 3 replicates, A1, A2, and A3. We are able to detect both of these mutations in 2 out of 3 replicates with a significantly better variant allele frequency. You can see in A1 replicate almost 50%, almost perfect 50% for both mutations. A3, you see the same. Here in A2, you can see that the percentage is a little bit lower. And keep in mind, we are not selecting specifically only tumor cells. We are selecting cytokeratin-positive cells. We try to purify it even more by using ploidy analysis to look for aneuploid cells. But of course, you will have some contamination by cytokeratin-positive non-cancer cells that may decrease it. But it will still take away all the fibroblasts, endothelial cells, and immune cells, so you're going to still be able to decrease it. What I want to point your attention to is not only variant allele frequency, which is significantly better, but that your coverage is no different than when you're doing bulk sequencing. So you see close to 2,000 read depth on bulk sequencing, close to 2,000 read depth on DEPArray isolated cells. And you can see that despite the fact that you use only approximately 0.6 nanogram of DNA input, for comparison, our clinical NYU Oncomine panel is validated for 10 nanogram original DNA input. By Thermo Fisher, you can go below 10 nanograms, but that's what we have in our original submission. But here you can see basically you can decrease your DNA input 20-fold by using pure population of whole cells. Let me show you quickly another example. This is a tumor that was estimated to be 15%. Obviously, it was not, because you can see variant allele frequency is about 55% here, EGFR exon 19 deletion, and this is how we selected the cells. Cell browser, you can see again FTSE positive keratin cells, very pleomorphic. You can see replicate 1, 2, and 3. Each of them we are able to detect exon 19 deletion with a good variant allele frequency, a little bit lower here, and again coverage is very good, close to 2,000. And that is using no modifications to our clinical Thermo Fisher Oncomine protocol. So you can basically take the protocol that you already have for the sequencing and process pure population of tumor cells. In addition, you can also find things that you didn't know that were there. So this is an example of a case that we knew had an exon 21 L855R mutation, and we knew it was variant allele frequency was about 21%. And then this is what it shows on the imaging from the DEPArray. You can see these very pleomorphic cells, strongly positive for cytokeratin. And you can see here, again, we do everything in triplicate because it was a clinical validation. You don't have to do it. You can see it's very reproducible. You can see that in addition to your L855R mutation, with a very good coverage again, we were able to identify EGFR amplification with 10 copies. These are your confidence intervals that come out. So you can see very, very reproducible results, both at the level of a single nucleated variant as well as a copy number. And again, to emphasize, we missed this EGFR amplification when we did the bulk sequencing, exactly as I showed you before. The number of cells that we used in each pool was 139, 153, and 145. So still significantly low number of cells that we needed for this analysis. So this was how we are using DEPArray in a clinical setting, and you can see it is very reproducible and a robust method that can be easily plugged into existing clinical workflows. In the interest of time, I didn't show you the results of the hybrid capture sequencing. You can still do it, but it does require more DNA than amplicon sequencing. So I'm going to show you just a few examples in the translational research that we are doing and cancer heterogeneity, which is the interest of my laboratory in particular, showing you some mechanism of resistance identification, and you can also use it for tumor stroma analysis. It allows a lot of interesting possibilities. So how do we dissect heterogeneity in cancer? This is an example of a glioblastoma infiltrating the brain. You can see a huge variability in a cell size and shapes. And when you do a bulk tumor profiling, you can see that we are able to identify multiple copy number aberrations. There is a MYC-N amplification, there is a PDGFR alpha amplification, EGFR amplification, CDK4 amplification. So that's great. You got a good profile. It's a very highly cellular tumor. You may think, well, I have a very good detection of copy numbers. I have a very good tumor cell percentage. There is probably very little need for me to do any additional analysis. But if you remember in my earlier slides, you have no idea whether PDGFR and EGFR are in the same cells or in different cells. And of course, that is an information that's interesting for us who study tumor heterogeneity. And when you optimize and validate your stains for these respective kinases, you can see that in this population, these cancer cells are strongly positive for CDK4 and PDGFR alpha, and when you profile them, they indeed carry only amplification of PDGFR alpha and CDK4. You can see that they still carry the same p10 loss that was identified here, so they can still come from the same clone. And then this other population that is highlighted here, it's positive only for EGFR. I'm showing here, for example, a channel which is negative for CDK4, and you can see that this sample has a trisomy chromosome 7, high-level EGFR amplification. It has a p10 loss here, but you can also see in here in the bottom that in addition to it has homozygous deletion of p16, CDKNA2A. And you can notice that this homozygous deletion was indeed missed in the original bulk sequences, because these cells that carry homozygous deletion were basically diluted by these cells that don't have homozygous deletion, and that's why it was not called. And of course, if you notice, you have PDGFR alpha, CDK4, EGFR. You're still missing a population of MYC-N amplified cells. So that is, there is at least a third population of glioma cells in the background that we need to identify. One of the reasons we're unable to find this population is because we don't have really good markers to pick it up from the background. Another translational research project that we are looking at is looking into metastases and genetics of metastases, particularly in patients who are undergoing immunotherapy. And this project originally came from the idea that we would like to analyze the immune infiltrate of metastases in patients who are treated with checkpoint inhibitors. But when we looked at biopsies of these liver biopsies, of these liver metastases, we were impressed by the fact that there was virtually no immune cell infiltrate. So that kind of bummed. Yes, there was, hypothesis was wrong. There was no immune population. But what we noticed is that in addition to these carcinoma cells that were pretty obvious, there was a very exuberant, what we thought is a fibroblastic proliferation in the background. You can see this myxoid stroma in the background, this spindle cells in some areas, probably more obvious than the others. They also are quite pleomorphic. And at that time, when we look at it first, we were like, well, there's a lot of mesenchymal cells, and they may represent the mechanism of resistance. Maybe these fibroblasts are somehow shielding tumor cells from immune cells to come in under the checkpoint inhibition. And this could be interesting fibroblasts just helping tumor to grow. So we were like, okay, that's going to be great. We're going to look at these Vimentin positive cells, so we're going to compare how they are helping the cancer cells. So the strategy was we're going to isolate keratin positive cells, mesenchymal cells positive for Vimentin, do next generation sequencing and some RNA profiling and see what's going on. So when we did our DEPArray experiment, we were surprised that in addition to pure population of keratin positive cells, which are shown here, and a pure population of small Vimentin positive cells, which are shown here, which we believe are really fibroblasts, there was also this rather abundant population of cells that were strongly double positive, both for keratin and for Vimentin. And they're also morphologically quite abnormal, or polyploid. And we realized that actually not two populations as we thought, keratin, Vimentin, but also there's a third population of these double positive cells. And we didn't know what these cells are. Are these fibroblasts that because of cancer are picking up expression of keratin, or are these cancer cells that are going through epithelial mesenchymal transition and they're starting to express Vimentin? So, the proof is in DNA. So we performed the sequencing with help of an analytical help of people from Menarini Silicon, and this is an experiment that was actually done using DEPArray OncoSeq sequencing panel, and you can see that the keratin positive cells and these double positive cells actually shared CDK4 amplification, and mutation is CDKNA2A, and particularly this amplification made us confident that these double positive cells are not fibroblasts that start expressing epithelial markers, but the opposite. They're actually cancer cells that started expressing mesenchymal markers. They look mesenchymal on histology, and they can be considered to be fibroblasts if you perform only logical evaluation, and they may represent an EMT as a mode of escape of immunotherapy, and this is an area of active research on our side, which we are now doing. So in summary, I hope I convinced you that because cancer samples are heterogeneous and tumor cell percentage and DNA input are critical to obtain NGS information, FFPE samples can be analyzed using DEPArray capture and digital sorting of whole cells. VRNxT is a new addition to a DEPArray workflow, and it enables rapid and accurate volume reduction. It prevents cell loss and limits operator variability. Next-generation sequencing can be performed on a small number of cancer and stromal cells to distinguish molecular drivers and identify subclones. Pure subpopulations enable us to better identify driver mutations, copy numbers shared in each subclone, and also pure subpopulations can help us identify subclonal mutations that we missed on bulk sequencing alone. And the analysis of cancer subclones help us to identify potential modes of resistance, and we are hoping this can be then implemented in a variety of different cancers and metastases. There's a large team of people who work with me. Some of the work that I showed you includes the clinical validation of the DEPArray, which is an NYU Langone Health molecular pathology team, sequencing core, collaborators at the Perlmutter Cancer Center here at NYU, and bioinformatics laboratory, my research laboratory, with many people who are pipetting and sorting and identifying cells. Some of these experiments were done in a very close collaboration with Menarini Silicon Biosystems, particularly in our early phases when we were learning how to analyze the data and how to properly capture the cells. And with that, I will end. I thank you for your attention. I'll be happy to answer the questions that are coming in.
Thank you very much, Dr. Snuderl. We have several questions that have come in, and they seem to be continuing to come in. Our first one, can this workflow be used on any type of cancer?
So any type of cancer is a broad term. So I would hate to say yes, any type of cancer, and then somebody will prove me wrong sooner or later. So far, it has worked on any type of cancer that we worked on. That includes primary pancreatic, the primary GI tumors, GU tumors, breast cancers, lung cancers, huge amount of work that we've done on brain tumors. I would say brain tumors are particularly tricky because brain doesn't have collagen and you need to do some adjustments in terms of enzymatic digestion. One thing that we are now currently working on is bone marrows. So I would say our experience with bone marrows is extremely limited, so I cannot really talk about it much in detail, but when it comes to solid tumors, I would say yes, pretty much everything.
Our next question, how important is the variability from single-cell heterogeneity in your liver metastasis research?
That's a great question. That's something that we're working on right now. Our original analysis was focused on small pools that range between 50 cells to 120 cells, and now we are doing a single-cell analysis to see how much variability is there on a single-cell level.
All right, a follow-up question to that last question. Is this variability correlated with increased chance of metastasis or responsiveness to therapy?
Yes, I would love to have an answer to that ready. The problem is that this was sort of a biased cohort because we specifically were looking at people who progressed on checkpoint inhibitors, on immunotherapy with checkpoint inhibitors, and our cohort is relatively small. So right now we are just looking into genetic and epigenetic markers of this specific population of patients that progressed. The reason why we went into DEPArray in the first place rather than just do bulk sequencing is because these scores were obtained under a specific clinical trial, and they were extremely small, thin liver cores that when we look at them, we just didn't think it would be amenable for bulk sequencing. And then DEPArray proved to be a very good way how to answer this question that we didn't originally even think there will be there about two different populations of cancer cells.
Will this work with slides?
I cannot answer that question very well because we try to recover cells from a 10 micron slide. We are able to recover some material, But because, as you remember my picture with the cells, how you slice them, you really get just the fragments of nuclei. So I don't think slides are a good starting material. I'm sure that somebody can make it work, but I think that really the advantage of DEPArray is to have as whole cells as possible. You can definitely get the material off the slides. You can stain it. You can load it on a DEPArray, but you will have to do a lot of manual selection because most of the things that are going to come from the slide are going to be judged by DEPArray as fragments. And those are usually not considered to be good objects for collection. You can manually collect them if you want to. So I think it's technically possible. I just think it will be a lot of work for not really good purpose.
Is there a limit in terms of maximum capacity of the cassettes to hold cells?
I think the limit is so there's a limit on how many cells you can load into a DEPArray cartridge. And one of my students will be probably better to answer that, but I think it's around 15,000 or 20,000 cells that you can put in into the cartridge, something like that. It's in tens of thousands, but not everything will be considered to be a good cell. There's a lot of fragments. Not everything will be possible to be moved around and recovered. So at the end, you end up with a couple of hundred of objects that are that you really are interested in after analysis.
Can DEPArray also be used for RNA-seq analysis for single cells from FFPE samples? And if so, what's the minimum or recommended input amount of RNA?
The short answer is yes, it can be, but it's extremely frustrating. And the reason is, so we've done the RNA-seq, single-cell RNA-seq analysis on cells from the brain, and our experience is limited, and we're still working through the data. And one of there are two factors. One of them is it is extremely dependent on the quality of your FFPE sample. And I would say that old FFPE blocks become almost impossible to use because you have so little. The second thing is the only thing that it is very difficult to reproduce. So if you remember the DNA data that I showed you, they were extremely reproducible. We did everything in triplicate. Every sample shows the same thing. And when you do DNA low-pass whole genomes, if you're able to pass the QC and you have a good QC result with 3 bands that Menarini recommends, your low passes are going to be good and you're going to be able to reproduce your data over and over. With RNA, there really, to my knowledge, there is no QC kit by Menarini. We don't have any good QC kit except standard RNA-seq QCs that these samples always fail, and then we did it anyway. And the only thing that we saw was able to predict whether our single-cell RNA-seq from FFPE cells collected by DEPArray was successful was the size of cells. We are able to get single-cell RNA-seq done on neurons, which are big cells, really big cells with big nuclei. We were not able to succeed with smaller glial cells. So that's sort of like anecdotal result that we are now trying to see. I mean, the results we get from RNA-seq are real. We saw that the genes, that are known to be overexpressed in neurons compared to glia were overexpressed, but it is very frustrating that your yield on single cells is going to be relatively low. Our strategy is to have at least 5 to 10 cell pool of cells. Like, for example, you take the neurons that are the same size, they're from the same layer, so you can judge them by the size and shape. And then if you have a pool at least 10 neurons, that actually works relatively well on RNA sequencing.
Anna Maria asks, have you ever done experiments on PBMCs? And if so, were there any particular problems?
I assume this is peripheral blood that you're referring to. Not on a peripheral blood, but we do a fair amount of work on circulating tumor cells. Some of the applications, and that's sort of the beauty of DEPArray is that you have one instrument that you can use both for your CTCs, which are fresh cells, as well as for your formalin fixed paraffin embedded tissue. So it works really well on CTCs. So I assume that if you're interested to capture peripheral blood cells, it would work as well. You need to keep in mind that how many cells you can load into a cartridge so you don't overload it, but it works very well with fresh cells, and there are other people, not us, who have done work with cultured cells as well.
All right. And this question, I believe you mentioned the answer during your presentation, but they're asking for clarification. How much starting volume is required to load on DEPArray?
This is going to be really embarrassing, but because I don't do these loads by myself, I actually don't remember how much it is, but if I remember correctly, it's somewhere between 10, 13, or 14 microliters. It's very important to have an accurate volume to not overload your chamber, but let's say it's 10, 12 microliters that you load in that. You do the cell count before, so you know how many cells you have in there, so you don't overload your chamber, and you have it in about those 12 or something microliters that you load in.
Okay, and we did get confirmation from Venice that it is 12 microliters. Our next question, for preparing the samples from FFPE sections before dissociation with the enzymes, is there a step for deparaffinization? Yes.
It is part of a protocol. It's regular deparaffinization with a xylene. So you need to have a xylene hood, but that's all what you need to do. It's pretty simple. If you're dealing with otherwise, if you know how to deparaffinize whole slides for any other method, it's the same. The one caveat I would make it is any other. It's obvious for anybody who does next-generation sequencing. Decalcified samples will not work with this technology, not because it's a problem of DEPArray, but because decalcified samples just don't work with next-generation sequencing well.
All right, and a follow-up question from the same attendee. For CK plus Viment plus cells, can they be a mixture of tumor cells and their partner MSCs?
Can they be a mixture? So that's a good question. We had a lot of discussion with the Menarini bioinformaticians. We tried to, when we created a pool, that pool was clean in the way that all the cells in that pool strongly overexpress both cytokeratin and vimentin when compared to vimentin-only population and cytokeratin-only population. So in that aspect, it's a clean population of cells. But if you ask me, is it possible that there's a mixture of fibroblasts that express cytokeratin and a mixture of carcinoma cells that express vimentin, I wouldn't be able to answer that without doing single-cell studies, which we're doing right now.
All right. Perhaps you can reveal those results with the next webinar. Our next question, can this technology be used to examine gene fusions?
We are currently validating whether, so for those of you who are familiar with the Thermo Fisher Oncomine panel, it has two components. It does sequencing both for DNA and RNA. So we are currently doing clinical validation of gene fusions present on Oncomine panel, but I don't have the data of that yet. So the honest answer is I don't know. And caveat is also we'll need to establish limits of detection, because while DNA QC-Kit is very good in predicting what is going to work for NGS, it's conservative. So if you're able to reach the number of cells recommended by Menarini QC-Kit, your NGS really will work. But we went down to like 30% of what QC-Kit required, and we were still able to get good results. On the opposite side, there is really no good prediction whether RNA will work. So we'll need to, if it works, I don't know. And if it does work, we'll need to establish limits of detection, what actually makes a good RNA sample.
All right. And time for one more question. Based on your experience, how much time does the DEPArray processing add on to a workflow up until NGS prep in a clinical setting?
So that's a great question, and it's a little bit hard to estimate right now, but my fabulous technician, Natasha, who is responsible for validation of that, is working right now how to make the workflow as simple as possible. It really depends how you're able to, how many samples you're able to process at the same time. What we are aiming for is that we batch these samples. You do preparation of let's say 4 samples at the time. You perform the deparaffinization, cell suspension, and staining at the same time. And then once everything is stained, sample is good for at least a week. The staining will be good. So then you can do one run after another and collect it. I would say it adds at minimum, you do suspension and deparafffinization and staining, and then run. if you time it well and you organize the work well, it probably adds at minimum 3 days before you can put the samples on the chip. The advantage is that what we are trying to do is that these samples will be just added to our regular workflow, so you don't have to run DEPArray isolated cells separately from the regular bulk sequencing, which is still, majority of our samples. We cannot do this on every sample in a clinical workflow, obviously, but we just add it on it. So to answer the question, I would say about 3-4 days of extra work before you can load it, before you can start preparing for NGS is about a reasonable assessment.
Thank you so much. I'd like to thank our presenter for today, Dr. Matija Snuderl, MD. I'd like to thank our sponsor for today, Menarini Silicon Biosystem, for sponsoring today's web symposium. Finally, I'd like to thank all of you for coming and spending this time with us. We know you're very busy, and we're very grateful you chose to come here, and we hope we provided some knowledge that will make life a bit easier for you in the laboratory. So on behalf of Cambridge Health Tech Institute's Global Web Symposia Series, thank you all so very much, and have a great day. Bye-bye.